is Nuraina. Tak jawab Nuraina. Oh Nuraina. Suddenly I saw your face. Nanti you share your fun facts, okay? You seem very focused on something. Okay, can you all see? Can you all see this one?
Yes, Doctor.
Yes.
Can okay, if I play, can you hear?
Acids. There are 4 special.
Can you hear the voice?
Yes.
Yes.
Right, good. Okay, let me know you have trouble. Okay, so I'll play the video at the same time. I stop and I explain. Yeah. Okay.
codons, 1 that codes for start and 3 that code for stop. Translation begins with the messenger RNA strand binding to the small ribosomal subunit upstream of the start codon. Each amino acid is brought to the ribosome by a specific transfer RNA molecule. The type of amino acid is determined by the anticodon sequence of the transfer RNA. Complementary base pairing occurs between the codon of the messenger RNA and the anticodon of the transfer RNA. RNA molecule binds to the start codon. The large ribosomal subunit binds to form the translation complex, and initiation is complete.
Okay, so when it comes to DNA translation, it's similar to DNA transcriptions, whereby you have initiation, you have elongation, and then you have termination, mula, tengah, dan ending. Alright. So when it comes to DNA translation, if you notice that you punya mRNA does not your start of your 5' does not mean necessarily this is ATG. So you have to remember, doesn't exactly that you the start of your mRNA, I mean the hujung, orang kata the pangkal of your mRNA, ending of your mRNA 5' is not necessarily is ATG. ATG could be downward, downstream, even 5 base pair or 10 base pair or 15 base pair, depending. So the moment it has some sort of ATG here, it will be detected. But you punya small ribosomal subunit, if you can look here, okay. So it supposedly, this is just a figure lah. Supposedly you punya small ribosomal subunit, if you look, it's like a palm, a bottom palm, and a big palm. Yeah, like this is not buaya, okay? Tolong don't be mistaken. I'm very bad with science. It's kind of like this. Yeah, big palm and small palm. Okay, small is kind of like flat. Okay, and then it kind of go, and then the big one is kind of go like that. Okay, so your small one is just floating in the cytoplasm. Okay, cytoplasm is actually a big, sort of like big vast of ocean. It's really big lah, comparatively to your punya ribosomal RNA. So basically, whenever it found mRNA, it sort of like binds. Tapi dia bukan bind exactly at this part, AUG or ATG part ni. Dia akan bind at the beginning part. So when it does, dia akan bind, dia akan hold first, and then it will start to scan until the moment, AUG lah, macam kenal je, right? Once it looks familiar, then another person will come in, which is your methionine, or the start of your codon, your tRNA. Like I said, always it starts with ATG, because this one can only be recognized by this one. So this one, if you look here, okay, so your coding is AUG or ATG, sama je lah. U is RNA. The anticodon, which is U, A, C, that brings the methionine. Okay, like I said, only methionine and another one is tryptophan are the one that only brings methionine. Okay, so that means the one that only has one codons. Okay, so once it recognize the scan recognize, now it become a tRNA and small ribosomal subunits. And then finally, bila ada this complex, all right, a bigger guy, which is your large ribosomal subunits, whereby it has 3 slots or 3 sites, which is E, P, and A. Nanti ada video show it to you. It recognize tRNA, it recognize the small ribosomal. Then it start to attach, become initiation complex. All right, this is a complex. Okay, so once it recognized, then it can start to translate the codons within the mRNA sequence. Okay, so that's the initiation. How all of those come together. All of those, your tRNA, your small ribosomal cell ini, your large ribosomal, dia bukan kawan, dia bukan gerak sekali It's separated because like I said, it's like macam you campak 3 leaves in a vast ocean. Atau dalam you campak lah you punya daun pokok or leaves, 3 leaves in a pool, they not always travel together. They just drifted away. But once it found sort of like a complex, they sort of attracted towards each other. That's how it works. Yeah. By the one that finds together, the one that find attract first is your small towards your mRNA. Then everything comes together. Clear? Okay, that's how initiation works.
The large ribosomal subunit binds to form the translation complex, and initiation is complete. In the large ribosomal subunit, there are 3 distinct regions called the E, P, and A sites. During elongation, individual amino acids are brought to the messenger RNA strand by a transfer RNA molecule through complementary base pairing of the codons and anticodons. Each anticodon of a transfer RNA molecule corresponds to a particular amino acid. A charged transfer RNA molecule binds to the A site, and a peptide bond forms between its amino acid and the one attached to the transfer RNA molecule at the P site. The complex slides down 1 codon to the right, where the now uncharged transfer RNA molecule exits from the E site, and the A site is open to accept the next transfer RNA molecule. Elongation will continue until a stop codon is reached.
All right. So this is called elongation, whereby, okay, you look here, okay. So you have this sites.
And A site.
Okay. You have E, P, and A. I'll explain to you what is E, P, A in the slides later. So the idea is the first one will attach at P, peptidyl transferase, whereby your tRNA, okay, actually bind with your amino acid through ester linkage. Ester linkage ni dia ada some sort of energy. Okay, if you still remember, Prof. Nazamin ajar, some sort of binding contain energy. The moment you break that binding, it release energy. You know what the energy is for? The ester linkage between amino acid and your tRNA. You know it's for what? Itu apa? Suka-suka je letak energy kat sini. What do you think happened afterwards? How on earth your amino acid is to be transferred to the next guy? Macam nak transfer here to here? Through the release of energy. Alright, so this one has ATP. What's the bond between amino acids to amino acid?
Peptide bond.
What?
Peptide bond.
Peptide bond.
Peptide bond. Good. So the growing chain, okay, from here the P site will be transferred using transferase enzyme. Through the breaks of transferase enzyme, it release energy that this will be transferred to the growing chain. All right. Once it grows, once it releases the amino acid, this one has no amino acid, become uncharged. Whatever tRNA that carries amino acid, that's called charged tRNA. Okay. So once it happens here, now you punya small ribosomal and large ribosomal start to slide. Okay. Once it slide, this uncharged will leave. Okay. This will come empty, allowing charged tRNA to come in. So only at A site, only charged tRNA would come in. If it's uncharged, it cannot come in. Because you have to understand, these are the bindings with codons and anticodons can happen anytime. Okay, because it's just complementary. Katakanlah ni, right? Yes, some codon. Sorry, katakanlah this one. Katakanlah this one is UUA, right? The complementary is what? UUA. Complementary dia is
AAU.
AAT. Sorry, AAU, correct. AAU, right? So any AAU that does not carry any amino acid, it cannot enter because that tRNA is uncharged. So that's how ribosomal subunit, small ribosomal subunit, control the elongation. Okay, if it's not controlled, any uncharged yang masuk, meaning that any tRNA yang tak bawa and doesn't carry any amino acids, so that would mean that this amino acid is transferred to empty one. Okay, maybe it transferred, maybe the whole thing just falls off because the next guy is empty. That will create a mismatch or misfolding protein. When it comes to misfolding protein, right? Any specific amino acid is different, or if a single one pun can change the whole dynamic of the protein folding. Nanti we discuss later in the next lecture. Okay, so having to have amino acid protein specific amino acid sequence is very important. Okay, so ribosomal are the one that control only charged RNA can goes in. Faham ya? And those that are uncharged immediately kicks off. It has to be kicks off. If it's not kicks off, then it won't create any slots here. Okay. All right, moving on.
Our uncharged transfer RNA molecule exits from the E site, and the A site is open to accept the next transfer RNA molecule. Elongation will continue until a stop codon is reached. A release factor binds to the A site at a stop codon, and the polypeptide is released from the transfer RNA in the P site. The entire complex dissociates and can reassemble to begin the process again at initiation. The purpose of translation is to produce polypeptides quickly and accurately.
All right, so basically this one, when it comes to termination site or stop codon does not have any tRNA. All right, these are the ones that does not carry any tRNA or tRNA couldn't bind because it's specifically reserved for release factor. This release factor is some sort of it's a disruptor that disrupt the whole binding between all of these complexes. So the reason why, because it wants to release the amino acids from the tRNA and become free polypeptide. Okay, so I said release factor binds here and then it disrupts the whole thing. Okay, by breaking the adjacent bond between all of these complexes. Clear? All right, and then finally, once you have polypeptide, this we learn after in the next class how polypeptide or protein, this particular polypeptide, is being folded into either part of protein or a functional protein, or protein that is combined with other protein. So you're gonna learn about primary structures, you're gonna learn about secondary structures, you're gonna learn about tertiaries, and finally quaternaries. All right, clear? All right. So let's get into slides. We can go briefly about the slides. Okay, can you see the slides? So in terms of translation, you have to have these are the key items: mRNA, your tRNA, and your rRNA, ribosomal. Okay, this is tRNA, charged tRNA. If it's charged, it cannot go in. So the function of tRNA transports specific amino acid to ribosome during protein synthesis. Each amino acid is recognized by one or more specific tRNA. We have about 31 tRNA. So it means that how many amino acids that we have? 20, right? So we have 20 amino acids, but we have 31 types of tRNA. So meaning that one amino acid might be carried more than one tRNA, except for methionine and tryptophan. Okay, so link amino acid together with tRNA using synthetase here with energy from ATP. So the one that combining amino acid and your tRNA is the enzyme synthetase, which carries a ATP sort of energy. So contains anti-codon, which is complementary to your codon sequence. Okay, so amino acid is at the end. So one end here at the loop here have anti-codon. Here it combines with amino acids. So you have to understand this is a rRNA. So this is a RNA sequence, a nucleotide based sequence. Okay, it's not a protein, right? So don't get confused. For rRNA, all right, consists a ribosomal RNA, means that a part of it, your ribosomal too, is a RNA sequence structures combined with protein. Okay, so when it comes to your protein, when it comes to structural protein within your ribosomal RNA, it is very important because when it comes to protein, it is a very, very specific side of biochemistry reactions. So when they form a complex, okay, that is very important because the binding is due to specificity of small and large ribosomal subunits. Okay, so it consists of small 40S and big 6S subunit. This is the sizes, total of about 80S when combined together. Okay, subunit are separated in cytoplasm but joined during protein, meaning that, like I said, small ribosomal unit atau large ribosomal unit does not always travel together within cytoplasm. Cytoplasm ni besar, right? Like I said, it's like a vast of ocean, seluas-luas lautan. It doesn't always travel together. But you have to understand, large ribosomal unit or small, both large and small ribosomal unit ada banyak. If you still remember the last lecture, it consists about 75 to 80%. Quality is like bersepah-sepah lah in your cytoplasm. So the chances of them colliding together or meeting each other is actually very high. The moment your small ribosomal site unit attached to your mRNA, tRNA comes in, there's like a close by of your large ribosomal subunit ready to join together. Okay. Okay. So it contains binding sites E, P, and A. So here 50 5,000 RNA bases, 1,900 RNA bases. Okay, so combine 49 different type of proteins, which is a lot, yeah. So this 49 types and 33 types of proteins. Okay, so in here, a combination of your RNA base and protein that shape like this. Okay. So small ribosomal cell unit with several initiation factor bind and scan for start codon methionine forming initiation complex. So meaning here, like I said, so your small subunit, the moment dia nampak RNA aja, okay, it ready to translate. It binds off. Okay, but you have to understand. I'm not going to go very detailed. Prokaryote is very simple. It just binds off like this. However, when it comes to eukaryote, it's a bit complex. You still remember your DNA transcription, whereby you require some transcription factors or protein factors. Like I said, helper protein for your RNA polymerase to bind. Transcription lah. Okay. Same as your translation. Your small ribosomal subunit ni for eukaryotes, high organisms, it may require some initiation factors like helper protein lah, but I'm not gonna go discuss in detail, just for you to understand that our system for eukaryote is not simple as it looks. It's a lot, macam kita ni for high organism, there's a lot of control because we are a very complex organisms or living organisms. We have a lot of regulation and control. For one simple mRNA saja untuk to be translated, ramai orang yang control cakap boleh tak boleh. Right? It needs a lot of help for the small subunit to be translated. Okay, but I'm not going to go to mechanism. It's a bit complex. Okay, it's not the thing that we want to discuss. We want to discuss about its relationship with food. Okay, so it form a complex. So here, the moment EPA, the moment your small subunit ni, they can scan until it found AUG. Once it found AUG, then this anticodon, which UAC, that carries methionine, can only bind, and your large ribosomal subunit can start to bind. So this is why having a start codon is very important. Meaning that only your translation and only can be initiate once you have this AUG. If you look it into your charts of your codons, okay, all of these CCG, UUA, this codon already is actually encode for certain amino acids, right? But your other tRNA cannot bind because your large ribosomal subunit can only comes in bila ada tRNA of this start, UAC, and small. That's how your protein your translation of your protein are being controlled. Clear tak? Faham tak? Okay, like I said, biarlah depan ni ada 10 pun, regardless if you have 10 bases or 20 bases here, all of these bases or codons will not be translated. This is what I call UTR, untranslated regions, or leader sequence. This one cannot be translated regardless. It can only be translated once it finds AUG. Okay, like I said, your large ribosomal subunit can only comes in once it detects small subunit together with tRNA. This one, then it will come. Without that, large ribosomal subunit, tak nak datang, tak cukup gang. I don't want to do it. So simple as a look, okay, may notice or you may not notice it, that's how your protein translation is being controlled to have a right sequence. So that's how you have a start codon. Okay. If not all this is translated, you may have a misfolding. Any misfolding is fine. Any misfolding is okay. Because I'm not saying it's okay. I'm saying that misfolding, the last thing you should concern is about it's being degraded. Okay. For misfolding proteins, sometimes it can lead to a certain diseases or certain unwanted biochemical reactions. For example, katakanlah, I would say
sickle cell.
Okay, yeah, good. Sickle cell. Nice, sickle cell. Katakanlah sickle cell, supposedly your protein, your hemoglobin is kind of, it looks like a concave Y shape, like macam piring, back to back, yeah? The shape itself is very conducive to carry oxygen and to carry carbon dioxide all at the same time, right? However, for sickle cell, it's kind of like flat. Yes, it can carry oxygen. Sickle cell ni one amino sequence saja berbeza. It kind of like flat. It can still carry oxygen, but not as sufficient. This is what I call that a misfolding, a functional misfolding. If it's non-functional misfolding, it's fine. Dia tak kacau any kind of biochemical reaction. That's okay. But those misfolding yang kacau the whole biosystem can lead to anemia sickle cell, which that person would carry very limited amount of oxygen. Yeah. So misfolding, the worst case scenario, it can lead to a lot of disease, genetic diseases, rare genetic diseases, Alzheimer, Parkinson, all these nerve degenerative diseases. Okay, that one is all from a misfolding. Okay. Okay, so your P site stands for peptidyl tRNA binding site. Your A site, amino acid tRNA binding site, and your E site is exit site. Okay, EPA. All right. P is for holding, A is for entering, E is for exit. So elongation, okay, each time a new codon 3 base is exposed by moving towards is known as translocation. Okay, move towards 3' is known as translocation. So it's allocate from one side to another. Sliding, I mean, I call it sliding of your ribosomal subunit along your mRNA. So a matching tRNA binds to the codon, so it has to have anti-codon. Existing amino acid chain polypeptide is linked onto the amino acid of the tRNA via chemical reaction transferase. Okay, so from your P side here, small, from your P side, okay, it transfer your, hold on, okay, this one, all right, this one. P side, a new guys comes in, it recognize your amino acid will transfer to your A side. A side is the growing chain lah. Okay, this one empty, slides off the E side, release, and then release. Okay, shifting one, the mRNA is shifting one codon over in the ribosome, sliding, exposes new codon for reading. Okay, it just work by sliding the sides. So translation, this is what for stop codon. Okay, when ribosomes hit stop codon UAA, UAG, UAA, or UGA, it won't encode for any tRNA. It only encode for a protein called a release factor. Okay, a release factor will bind instead of tRNA, and it will add water for hydrolysis, causing the whole complexes to breaks off from each other. Okay, mRNA breaks off, ribosomal breaks off, and finally, most importantly, your polypeptide. What we want is the products. Okay, all this will require an energy, hydrolysis lah, water, and then everything will be, the break of all the binding complex will require energy through hydrolysis process. Okay, so here polypeptide may still need to fall into 3D shape. We learn in next week class. Okay, so the messenger RNA can be translated many times to produce many protein copies. Okay, like I said, if you look into last week class, I mentioned to you about feedback inhibition, or positive and negative. As long as that mRNA is required to produce that specific enzyme, it will recycle every time. Okay, the moment that mRNA is not required anymore, it starts to be degraded. Because as long as your mRNA ada in your cytoplasm, your ribosome will automatically translate, unless ada macam sort of repressor lah that binds to the start of the mRNA that will not allow your ribosomal subunit to be attached. But as long as it free roaming in your cytoplasm, it will be translated many times. Okay, for example, katakanlah kita your insulin cases. Okay, the moment we consume glucose or sugars, insulin will continuously being produced to convert the excess sugars into glycogen. So the moment your glucose is finished, no more lah, cukup lah, tak payah produce insulin anymore, right? Because there's no glucose is being there's no more glucose is being converted, is wanted to be converted from glucose to glycogen. So the mRNA responsible to make insulin will be degraded. Example, okay. And another one whereby example, your mRNA will be stopped based on the finished products. This is called negative feedback inhibition, like ATP. ATP is the final product, yeah. ATP will inhibit the initiation process of producing ATP. Right? The moment, for example, katakanlah when you are going to sleep, right? When you're going to sleep, you most likely will use lowest amount of energy as compared to your running. Okay, when you're sleeping, the accumulation of your ATP, which is your ATP is the final product, that product will some sort of inhibit or repress the production of ATP. Okay, so the mRNA responsible to convert your glucose or your glycogen to ATP will be stopped, right? However, the moment you wake up and then the moment, oh, nak wake up, nak pergi jogging. At the time you jog, your ATP is being depleted. So insufficiency ATP does not have, the insufficient ATP will not stop you punya initiation process of your making more energy. So at that time, enzyme that is required to burn your glucose to produce ATP is turned on. That's called negative feedback inhibition. At that time, your mRNA will not be degraded. It can be used many, many times as long as it's still being required. Clear, yeah? Any questions? All right, good. So we have 20 amino acids and then we have 64 combination of codons, okay? It's 4 to the powers of 3, okay? 4 type of bases, 3 codons, 3 places of codons, okay? Due to degenerate code of amino acids, right? Some tRNA can recognize several codons because the third spot can wobble or be mismatched, okay? So in terms of wobble or mismatch, yang hujung-hujung ni dia goyang sikit. Dia macam dia relax sikit. It's not that strict, yeah. Meaning that tRNA can be leucine amino acids can bind to different codon on RNA as long as minimum 2, the first 2 bases fit. The third one ni taklah specific sangat. It's fine. That's why when you look into the table of your amino acid with codons, some of it, especially the third part, slightly not exactly specific. For example, this one, you see dia punya mRNA is CUC, by right is to be GAG, normal pairing. However, suddenly your mRNA is CUU, but the one that carries the leucine, GAG. GC match AU match. The third one ni is wobble. It doesn't really macam require very specific bindings. 2 out of 3 can allow this leucine to be bind to the mRNA, right? So allow, therefore only you have 31 tRNA that carries 20 type of amino acid for the 61 codons. So my question would be, kenapa 61 out of 64? Why we only have 61 working codons? Kenapa?
Start codon ke doktor?
Is it because we minus the stop codons?
Correct. Because the stop codon does not carry any tRNA. How many stop codon we have? 3. So 3 minus 64 would be 61. Okay. Only 61 codons that encodes for 20 type of amino acids. Okay. So my question would be, why the wobbling or mismatch exists in terms of translation? Why this thing exists in terms of wobbling?
To like make the protein detach, I think. Is
Okay.
it correct?
Kinda, but not the thing I'm looking for. Any idea?
Nope.
So when you see this, okay, this one, kalau macam in terms of your RNA polymerase, right, ataupun you punya DNA polymerase, specific, right? Kalau A mesti dengan T, G mesti dengan C, regardless, it cannot be mismatch or wobble. That has to be specific. But why not this when it comes to translation of protein? Kenapa? Any idea?
Is it as the release factor?
Is what?
Is it as release factor?
Reduced spectrum?
Release factor?
No, not really. The release factor is for the 61 codons. My question is, why the wobbling or mismatch exists in terms of translation? Because if you have learned up until now, when it comes to DNA structures, replication of DNA, transcription, all of that is, we're talking about specificity. A dengan T mesti always together. G dengan C mesti always together. But kenapa in terms of this, it can have wobble pairing? Any idea?
Is it maybe because then we would need to use less tRNA?
What?
Is it maybe because we to translate the mRNA we would like require less tRNAs?
Yeah, kinda. So if you look at a number, we only have 31 tRNA. We don't have 61 codons. We don't have any 61 tRNA, right? So some of it may have to be macam bertolak ansur, they have to share in terms of codons. So this scenario create what we call flexibility. Have to have flexibility because the tRNA is not enough for each of these 61 codons. Unless we have 61 tRNA that fits for 61 codons, then it's different lah. But the matter that the fact that we only have 31 combination of tRNA that carries 20 type of amino acids, so it has to have some sort of flexibility of wobbling so that this tRNA can bind to or can accommodate to 61 codons. Faham tak? It's like macam you have 31 food for 61 people. How? Right? How? So this is whereby some of you guys needs to learn how to share. Some get one because maybe they're a bit manja sikit, but the rest might get like share with 2 people, share with 3 people. So decrease the flexibility is important because we don't, if it's too specific, meaning that some of the codons, some of the DNA cannot bind with your mRNA. So when it cannot bind, this would leads to empty codons. If empty codons, that will lead to misfold juga. Okay, because if it's empty, no tRNA can come, then habislah. How do you going to fold? So in terms of transition, there exists some sort of flexibility due to wobbling or mismatch, wobbling effect. Faham tak?
Yep.
The idea is you have 31 tRNA to fit or accommodate 61 codons. So some of them have to share. The sharing will create a flexibility so that continuous production of a continuous bindings of your amino acids towards the growing chain. Right? If it's too specific or too rigid, okay, dia ketat sangat, tak ada flexibility langsung. So there might be a place or codons that none of the tRNA could bind. Make sense? Clear up?
Yeah.
All right, good. Okay, prokaryotic and eukaryotic. Okay, prokaryotic, it has no cap in terms of mRNA. No cap is no guanine cap. Has specific ribosome binding site upstream of AUG. Polycistronic, multiple protein from the same RNA, meaning that it can, one mRNA can have literally like 100 people working on them. People to mean ribosome lah. Okay. It transcribe and translate. Sorry, it translate all at the same time. Okay. So in terms of this, they are very efficient at the same time. That's the reason why your bacteria has a very high growth rate or high cycle rate. Okay. Meaning within a day, within 24 hours, anak dia dah beranak like ada 10 generation dah. Okay. For humans, for us to have one generation, it takes about 20 to 30 years. Or you get like, for it to get married, to produce kids, like 20, 30 years. Right? For them, a day. That's why when it comes to eukaryotic, not like talking about humans, like animals or plants or be it your fungal. Okay, fungal, the lifespan is about 1 week or so, or maybe months. Hardly will be a days lah. The maximum I've seen, like the shortest would be like 3 to 4 days for them to start to grow. Okay, because they are very complex. There's a lot of regulations are being controlled within eukaryotes. So eukaryote is monocistronic. One polypeptide per RNA. Once one polypeptide is finished, then it can be recycled to make a new polypeptide. recycle again. So prokaryotic tak payah tunggu. Anyone can attach. Ambillah korang satu ni. Buatlah ramai-ramai. Lots of workers, ya. Okay, eukaryotic transcription occur in the nucleus, in eukaryotes, nucleus in bacteria, open space for bacteria. Translation occurs on ribosome in the cytoplasm. mRNA is transmitted outside the nucleus through nucleopores. We learn about this. Okay, the difference between eukaryotes and prokaryotes. Okay, this is sama. I think I explained it before. Why is it that when it comes to prokaryotic, it's polycistronic, allow very fast translation? Because, like I said, it's to accommodate their high growth rate or their high cycle rate for prokaryotics. Okay. When it comes to prokaryotic, sama juga for eukaryotic. This is an example of prokaryotic circular DNA. Okay, you have your 5' and then you have 3'. Okay, I'll teach you how to learn the gene. Okay, before this, we understand that 5' to 3'. Yeah, we understand that A, T, katakanlah this one A. Let me, okay, here. I'm not sure. If I scribble, it's gonna be hang. Just bear with me. Yeah, if you don't understand, just ask me a question. Okay. So the direction, okay, let me just scribble je lah. Okay. So we understand that all this while is go from 5' to 3'. Your A, T, oh, okay, cantik kak? Right. ATG dot so dot your the rest of your nucleotide sequence is being translated. Okay, this is your coding. Katakanlah this is your coding sequence. The complementary genes of this sequence will be template sequence, whereby your RNA polymerase would bind. All right, clear. However, realistically and in reality, it does not always headed from 5' to 3'. It could be although it's 5' to 3', the genes is read from here to here. How's that even possible? Yes, it's possible because you punya DNA, you punya RNA polymerase, it does not have to have ATG as its binding site. All it requires is a promoter sequence and your termination sequence. The termination sequence does not always parallel with your mRNA. So meaning if the gene A here, meaning that dia punya coding sequence would be A, T, G. The rest is just follow. Okay. So your RNA polymerase will bind here because it's coding sequence, and it starts to produce your mRNA nucleotide sequence, and it has one of the sequence will be GTA because RNA polymerase can only bind from 3' to 5' to produce 5' to 3' RNA sequence. Clear tak? Does it sound confusing to you? Faham tak? Hello?
Faham, Doctor. You're just
Anyone's
saying
not
that
clear?
you're just saying that it doesn't need to have a specific direction, right?
Yeah, the direction doesn't matter. It's like crossword. If you have done crossword, katakanlah you spell for you spell for oh my god, okay, you spell for biochemistry. Okay, the normal way we write biochemistry is from left side to right side, betul tak? But in crossword, as long as it's spelled biochemistry, it doesn't matter if you go from right to left, down to up to down, diagonal, that way or that way. Doesn't matter as long it spells for biochemistry. The direction doesn't matter. Okay. When it comes to RNA polymerase, like I said, RNA polymerase does not require the recognition of ATG. RNA polymerase dia require apa?
promoter and
Exactly. Promoter
exactly?
sequence and termination sequence.
Termination.
Yeah, that's all it requires. Where and from what doesn't matter. For example, this gene is heading toward that side. Okay, your RNA polymerase bind kat mana? Sini, right? Betul tak? So it translates from what, where to where? Your RNA polymerase from here or from here? Which one? For gene B, ke kanan ke kiri?
To the number 5.
Towards the number 5. Okay. So when it comes to translation, all this, always remember the rules of, the rules of transcription. You always have to remember, it always follow the rules of 3' to 5' on the template sequence. The direction of the gene doesn't matter. All you have to remember the basic rules. Okay, it has to bind 3' to 5', and it produce 5' to 3' RNA. Faham tak? Clear? So don't get confused. So this is what we call what? Sorry. Undo. Okay. So this region is what? This region is what? What sequence? Template, not template, coding? Hello? Guys?
Template strand. It's 3 to 5. Right.
No. The template
Okay.
strand is here. This is non-template strand or coding sequence. Jangan confuse.
So there must be, and the non-template strand must be 3 to 5.
No, doesn't matter. Template strand could be 3 to 5 or 5 to 5, doesn't matter.
So you're saying
The
that based
template,
on
okay, the basic rules of template or non-template, if the coding sequence is here, okay, now ini part direction of the gene, right? Means that if it's at the bottom, okay, and this gene C, means that this one, the bottom part of the double-stranded DNA, this is the coding sequence. Doesn't matter if 5' or 3' direction. Like I said, crosswords. Yeah. But this is coding sequence. The rule is this is coding sequence. Automatically, this would be the opposite or complementary side would be template sequence, where your RNA polymerase will bind. Clear tak?
so the RNA polymerase just follows the direction of the template strand like that.
Yeah.
Okay.
Clear tak?
Clear. For me lah.
This is my favorite question ni kalau answer ni. So better be clear. Any questions before I start to move on? All right. If no question, I'll assume everyone understand lah. Okay. Clear. All right. Move on. Okay. Finally, once translated protein are finished, they must undergo post-translation modification. You have to have correct folding, addition of functional groups, building subunits together of protein with contouring structures, and cleaving apart of protein. So basically, your protein keluar ni macam burger flat macam ni. It's really unappealing as it doesn't function well. Whereby after the folding, adding functional groups, building subunit together, combine with different protein, or you cut parts of protein, then become nicely functional burger, which is not ideal lah. This one doesn't exist unless you make it on your own. Okay, but the idea is you make from protein, you fold it, you make it into nice form, working form, then it becomes fully functional protein. Okay. Tengok burger lapar lah pula. Alright. Okay, modification. Life affects the lifespan, cellular location, and activity. Okay, this is going to learn about the protein later on. How some of the protein is being added a few functional group that makes the lifespan of shelf life much longer. Some protein needs to be modified so it can enter different spaces of the cell. And then some protein needs to be modified so that it serves different kind of activities. We have a lot of different activities. Some is for digestion, some is to produce energy, some is being used as a waste management, all different kind of things. Okay. Okay, let's discuss together. I think this one you can recap on your own. Do you all have any questions? Ada soalan tak? A very quiet class today. I assume semua faham. Just ulang banyak kali kan benda sama. I'm sure it's very easy for you guys. Or is it too heavy for you? Okay tak?
I think slowly digesting, sir.
Slowly digesting.
processing.
Okay, faham sikit lah. Nanti adalah tanya soalan dalam WhatsApp tu. Sekarang tak tanya. Kan dalam WhatsApp kan macam jailer tanya. Bila nak exam lagilah banyak tanya. Biasalah tu. Dekat nak exam baru kelar kabut. Ni tak faham. I understand what is this, just ask me again. Pancung you guys. Alright. So, okay, I need to recap. This one you can recap on your own because we don't have much time. Okay, don't forget your there's 2 assignments, sort of like short assignment that I gave it to you. Your short nucleotide sequence. Alright. And the other one whereby I want you guys to come up with feedback inhibition on your own. Ingat tak? Bapak lupa lah, Faris. Apa dia? Tanya group balik. Rachel, you still remember at Rachel?
Yeah, doctor. I sent to the group.
Then you all have to remember. If she doesn't remember, I'll remind her. So we have 2 things that I supposedly I want to discuss today, but since we don't have much time, so the first one would be the single nucleotide RNA, or we talk about the introns, the exon, remember? Introns and exons. What's the function of introns? Kenapa they exist? Okay, you find out about that. And then another one whereby I want you guys to come up with the biochemical process or biochemistry process or reactions that involve either feedback inhibition or negative feedback. Sorry, positive feedback inhibition or negative feedback inhibition. You can just Google it. So since we are unable to discuss right now, can you all put into assignments your name or your matric number? Rachel, can you just post it to the group, the questions? So
Yeah,
everyone
I can,
has the same
sir.
question. Okay, you can send it to me first for me to check whether this question is right. Send it to me first. Then if I say it's okay, send it to a whole class. All of you use the same question. Answer in you can use a PowerPoint or you can use a Microsoft Word. Doesn't matter. And I'll create a link. Rachel, please remind me to create a link for next week.
Our classes, sir.
Remind me again on Monday.
Okay.
Create a submission link. And another one would be, I think I've mentioned to you guys about the YouTube link, the genetic modification and CRISPR link. Remember? Okay, please watch those YouTube. If you want to watch more than the link, up to you, it's even better. I want you guys to come up with a application or potential application that people have done using GMO to enhance food, to make food safer in terms of food punya quality, food punya nutrient, food punya safety, those kind of things. Like for example, nowadays they produce a bigger pumpkin. Pumpkin, they're able to have bigger size. Or nowadays they produce a certain genetic modification to have much more red color apples. So red color tomatoes. It's more appealing. And then some of the corns they produce nowadays that have natural pesticide resistance. Sorry, natural pesticide. Meaning that before this, orang kena sembuh racun perosak, pesticide or herbicide. Okay, to avoid any insect to destroy the plants, to destroy the corns. Now, scientists have created a gene, scientists have found a gene that is responsible to produce a pesticide and they insert into corns. So that corn punya plant produce naturally the pesticide. The moment insect makan that corn, dia mati. But it's safe for humans lah. So meaning that nowadays farmers, they don't have to use pesticide spray anymore. Biar je macam tu. Insect makan, makanlah. Mati korang, sesepah mati kat tanah. So that's the one idea of the use of genetic modification. Find it, Google it. Okay. What I want you to do, okay, next week class, I'll have 1 hour. I think I'll finish the next class in just the next lecture in just 2 hours. Right, I make it nice and short. And then the remaining one hour class on Friday, I'll select randomly. Yeah, randomly. Siapa-siapa yang senyap, tak keluar video kamera tu, kenalah. Randomly, doesn't necessarily korang-korang bisi-bisi tiba hari Selasa tu, oh, havoc. Semua nak jawab. To be safe on Friday lah kan? No, not really. I'll pick randomly juga. So regardless, korang bising ke, korang havoc ke, tak havoc, kena juga. So basically on Friday, I'll have 1 hour class for us to discuss, to share with you, for everyone to share your findings. Let's make it interesting lah. Okay. So you can do it in, if you want to make it more interesting, do it in PowerPoint slides, where I want you to present in a very most layman's term. Kiranya kalau you explain kat mak you pun, mak you faham. You can use figures in a very simplest way you can explain. I don't want technical as in masuk gene coded for GEN 13P4P. I don't want to hear all that. All I want to get is just a brief idea how genetically modified food or genetic engineering or CRISPR-Cas9 have affected or produced some sort of enhancing our food. Clear? It's just in the layman form, kalau ada figures banyak, like from here, from A to B to C to D. That's how it works. Cukup. Like I said, if you explain to your mom or your grandmother, they will understand. In a layman's term. I don't want you punya kerja like PowerPoint bombastic, like, oh, very detailed, it's for this research, that research. But when I ask you certain question, you would understand. You just baca copy je. I can easily detect that. So no need to do that. Baca, fahamkan, explain in a very most easiest way. 5 minutes, cukup per person, just share how genetic engineering has affected human life in terms of food production. That's it. Okay, in the most simplest way. Tapi janganlah simple sangat macam kerja tadika pula. Okay, so there's a fine line between very simple and very complex. All I want you to do for you to understand, do it in a PowerPoint slide. One single PowerPoint, single slide pun dah cukup dah. You don't have to be like 10 slide. Okay, saya hari ni nak beritahu korang 10 slide. Kau, dia lah bagi lecture. Right, don't need to do that, right? Kalau nak jadi lecturer kan, later on lah. If you want to become lecturer, then you can apply for lecture job later on. Understand? Faham tak? I'll pick randomly, so make sure everyone, they must have their work. Yeah? Because this one has marked lah. If you don't present, if Sunday, oh, alamak, tak ready lagi, kelang kabut nak cari at that time, then you see the other side of me. Okay, let's hope that will not happen. Okay, so everyone, please be fair to each other. So if one person does a job, everyone does a job. Clear? Any questions about your slides or your presentations? Simple je.
Doctor, yang presentation tu is about the food yang by GMO in food kan?
Yeah.
Yang the other tu yang introns and the
that one is just submit je. Nanti I create a link.
Submit. For the introns kan, doctor, I think like bukan tak ada macam specific function lagi for those introns. Like banyak in research kan?
Okay, if you Google online, scientists have not actually find out the function of introns. It's macam dulu don't ever find out the use of appendix. But they have some theories. You can Google online. They have some theories whereby this intron is being converted into snRNPs. You can Google snRNPs.
So kita just cari any like facts yang plausible lah.
Yeah, possible. You can share with the info with the whole class. I mean, you most likely you will have the same answer, but I don't expect you to copy and paste each other's work lah, because your style writing is different, your understanding is different. You can discuss with the class the function, but the way you put into your assignment, because I'll check, yeah. Don't expect I won't check. I'll check, and then your ideas might be different. The way you writing will be different. The way you conceive it will be different. But of course, you can discuss with the whole class in your WhatsApp group to share knowledge.
We submit in like PowerPoint or Word. It's up to us, right?
Yeah, it doesn't really matter.
Okay.
Doctor, where's the deadline for this assignment? The 2 questions.
Everything will be next the submission will be next Friday, 11:59 p.m. So 1 week from now, should be plenty of time for you guys to do it. So I'll give you 1 week, right? You can just finish it within a day, less than a day, half a day, 2 hours tops. And please do not. I want to emphasize very strictly. Do not, and I say, do not copy and paste and plagiarize. Once I caught, because I know the way students are writing, kalau you tulis cantik sangat, your English is perfect, it's really sound like a paper or journal or something like Wikipedia. If I copy and paste that, you can use the software to detect you punya assignments. If I find out there's a copy and paste, you will be very, very big trouble. So I'll tell you now, and I tell you directly, do not. If you want to use that words pun, please lah, tolonglah jangan malas sangat. Paraphrase lah. Okay. Copy other works is fine. Bukan copy, refer other works is fine. As long as you cited and then you give credit to them. Macam lab report lah. Okay, I'm sure you've learned this. Do not copy and paste and plagiarize. If I caught you, you'll be in trouble. Simple as that. Okay. So all I want to do is to be fair. Ada orang lain, your classmates, buat kerja penat-penat, cari semua all the resources in the internet, use not copy and paste. So it's not fair. Okay. So I'm just speaking on that. Clear, yeah? Just want to be fair with everyone else. All right. Any question? If no question, you all can go to lunch. Oh, suddenly ramai orang turn on video camera hari ni. Good job. Aina kata nak share fun facts. Tak nak dah?
Ya.
Aina, okay. Aina ada, okay, dua Aina. Dua. Aina dengan Nuraina. Korang dua lah share fun facts.
Nuraina. Panggil saya Nuraina.
Okay, panggil. Macam marah je. Sorry lah, Nuraina. Sorry, macam marah je. Panggil saya Nuraina. Sorry lah kalau saya lupa Nur di hadapan. Nur tu kan cahaya. Nuraina. What's your fun facts? Come before we leave.
Tak fun pun, tapi I just found out that amylase, enzyme amylase tu is the first proof yang dalam badan kita ni ada chemical reaction.
Ada what?
Chemical reaction.
Chemical reaction. So they discovered amylase is the first enzyme. Oh, okay. Yeah, that's fun. Fun lah juga. Okay lah, fun lah. First quote. Okay. Aina?
I think it is out of the box sikit.
That's fine.
Tapi I just found this. It's about
did you Google?
dolphins.
What about dolphins?
It says that dolphins have been witnessed entertaining themselves by creating underwater bubble rings.
Sorry, what?
Dolphins have been witnessed entertaining themselves by creating underwater bubble rings.
So dia main di sini dengan buat bubbles.
Buat
Yeah.
dalam air. Kita
Yeah,
bersama.
macam bosan-bosan. Bosan-bosan buat bubble.
Cuba manusia bersama. Cuba korang masuk dalam air buat bubbles. It's like entertain, doesn't it? You see your own bubbles. Alright, okay. Ati, thanks, Aina. Okay, anyone else? If not, we just end our class. Thanks, Aina. That's a very fun fact. They know how to entertain themselves. Kita buat apa? Alright, maybe you all watch YouTube or something. Alright. If no questions, if nothing they want to be discussed or questions, I'll end the class. Yeah. Thank you very much, guys. You all have a good weekend. See you.
Thank you, Doctor.
Doctor.
Thank you. Bye bye.
Thank you,
Thank you,
Doctor.
Doctor.
Thank you, Doctor.